Hello everyone. Today we are solving this GATE 2022 question from the paper Life Sciences, question number 64. And my name is Shurosri Pal. I am a Prime Minister's Research Fellow in the Department of Biotechnology of IIT Kharagpur. Now let's see what the question states. When a true breeding tall plant containing red flowers was crossed with a true breeding dwarf plant. So I would underline these things here. Tall plant with red flowers and also true breeding. It was crossed with true breeding dwarf plant with white flowers. All F1 plants were tall with red flowers. Tall with red flowers. When the F1 plant was self-pollinated, considering independent assortment of plant height and flower color traits, the calculated percentage probability of dwarf plants bearing red flowers in the F2 generation is how much percent? And we round off the answer to two decimal places. Okay, so this is a question from Mendelian genetics, and this is also a question that is concerned with dihybrid cross. Why? Because we are here considering two characters. One is plant height, another is the flower color. So there are these two characters, plant height and flower color, and we have to determine the percentage of dwarf plants with red flowers in the F2 generation. So this is a classical Mendel's dihybrid cross. And if you have gone through the Mendel's monohybrid and dihybrid cross and the laws that Mendel stated out of the results of these crosses, uh, we will just briefly overview the Mendel's dihybrid cross. So this is the Mendel's dihybrid cross. So dihybrid because it involves two characters. So in the Mendel's dihybrid cross, we know that the characters were seed color and the shape of the seed coat. Got it here. Seed color. There were two characters, seed color and the shape of the seed coat. Now these characters again had two alleles, two different alleles. So seed color was either yellow or green, and the shape of the seed coat was round or wrinkled. Now the dominant character among these were known. So the dominant character in the seed color or the dominant trait in the seed color was yellow. So yellow was dominant over green, and in the shape of the seed coat, the round trait was dominant over the wrinkled seed coat. So round is dominant over wrinkled. So that is why round is denoted as capital R and wrinkled as small r. Whereas yellow is dominant over green. So yellow is denoted with capital Y and green with small y. And it is very critical to note here that the dominant characters are usually uh, denoted with the capital letters. Okay, so now in the FP parental generation, Minden selected these two plants bearing yellow and round seeds and green and wrinkle seeds. So these two plants, they were true breeding. So this is something mentioned in the question, true breeding. So what does true breeding mean? True breeding means they are homozygous for both the characters. So these plants are homozygous for yellow and round and green and wrinkled, right? So capital Y, capital Y, capital R, capital R, it was crossed with small y, smaller, smaller. So these were the parental plants. In the F1 generation, these plants led to only one type of progeny, which is its phenotype is yellow and round, but its genotype is a heterozygous type. So it is heterozygous because it has capital Y, capital R smaller. Now, so it has basically the alleles, the round and wrinkled both the alleles, and yellow and green both the alleles. But since yellow is dominant over green, so only yellow color is expressed, and since round is dominant over wrinkled, so only the round seed coat shape is expressed. So all the F1 progenies were yellow and round, but they were heterozygous genetically. Okay, so now when these F1 plants, when they were self-crossed, so when these F1 plants were self-crossed, which means that this was crossed with the same type of, so the same genetically same plant. Then what happened is that these gametes, we know the gametes of this F1 generation, it produces four different types of gametes. So that is what was explained by the Mendel's independent assortment, Mendel's law of independent assortment. And he stated that, so this I will just discuss a little bit. During gamete formation, the law of independent assortment is stating that during gamete formation, the different pairs of alleles segregate independently of each other. So if there are two pairs of alleles, so for example, here in the parent cell, you can see there are two types of alleles, A and B. So these pairs of alleles, they will segregate independently of each other. So the segregation of one pair does not affect the segregation of other, which is why we can see these gametes which are produced in evenly in meiosis, which means that the probability of these gametes are all same. The probability of production of all these gametes are same. So capital A can pair with capital B, capital A can pair with small a can pair with capital B, small a can pair with small b. So all of the combinations that can arise from this gamete, all of them are probable if the genes are independently assorting. And this independent assortment is inhibited by linkage. So if there is any linkage between the genes, then this independent assortment does not work. However, for the if we consider the Mendelian genetics, actually Mendel did not have any idea of this linkage, and luckily enough, the genes that he selected or the characters that he selected for his experiments, they were not linked characters. So, which is why he did not observe any linkage between the characters. So, uh, according to this law of independent assortment, which was given by Mendel, we can see that these four types of gametes, they form independently of each other, and they all form in equal probabilities. So now, when you draw this Punnett square, you can find out that these are the progenies that generate in the F2 generation, and what will be the ratio. So the phenotypic ratio of this will be 9 is to 3 is to 3 is to 1. 9 yellow and round. So the yellow and round, which is the parental type, that will be produced in the most numbers, which gives the ratio 9. And this 3 stands for the green and round. This is yellow and wrinkled. And the minimum number of progenies will be the recessive type, which is the green and wrinkled. So that will be your um, dihybrid ratio of Mendel, the phenotypic ratio 9 is to 3 is to 3 is to 1. So I'm sure you are well acquainted with this ratio. Now, if we go to the question is stating that there is a true breeding tall plant with red flowers. Now, if we denote it uh, with the characters and then we can denote this tall character with capital T because why? Okay, now before doing that, let's see. The true breeding tall plant with red flowers was crossed with true breeding dwarf plant with white flowers, and all F1 plants were tall with red flowers. So considering the Mendel's dihybrid cross, what would we see in F1 generation? In F1 generation, we saw only the dominant phenotypes, which was yellow and brown. So in the F1 generation, whatever characters we can see in the F1 generation, those will be the dominant characters. So therefore, in the F1 plants here, according to the question, we see these characters tall and red flowers. So they will be the two dominant characters, and we will assign them with capital letters. So tall we can assign with capital T and red we can assign with capital R. So I guess you are uh, you understood this quite well that why we are considering tall and red to be dominant because we can observe these phenotypes only in the F1 generation and considering the dihybrid cross of Mendel, we will only see the dominant characters in the F1 generation. So tall is dominant over dwarf. So that is why dwarf we consider as small t and red is dominant over white. So white we assign with small r. So these are the characters. And when the F1 plant was self-pollinated, there was independent assortment of the plant height and flower color traits. So it is mentioned here that there was independent assortment. So we are not considering any chance of linkage in this particular scenario. Okay, so now let's see what happened till F1. So if we draw it here, the tall plants with red flowers. So this is the generation P was crossed with dwarf plants with white flowers, right? So now what will happen in the F1 generation? What will be the genotype? The genotype in the F1 generation will be capital T small t, capital R small r, and the phenotype will be sorry, the phenotype will be tall and red flowers in the F1 generation. Now, the next part is that now these F1 plants were self-pollinated or self-crossed. So now if you self-pollinate them, that means what will happen in the F2 generation. So in the F2 generation, if we now consider the phenotypes, what will be the different types of phenotypes that will be generated in the F2 generation? So because it is asked in the question that the calculated percentage probability of the dwarf plants bearing the red flowers in the F2 generation. So a particular phenotype is given, dwarf plants with red flowers, and we have to calculate the percentage probability. So where we will get this percentage probability? Obviously from the Mendel's dihybrid ratio, the dihybrid phenotypic ratio 9 is to 3 is to 3 is to 1. Now let's see what will be the phenotype in these F2 generation in this particular scenario. So, sorry. So now there will be four types of phenotypes that will be produced, and they will be first of all tall and red flowers, which is the parental type, and this will be 9 in the ratio 9, followed by tall and white flowers, which will give the ratio 3. Dwarf and red flowers. So we are making all possible combinations that can arise. And the least probable one, which is the dwarf and white flowers. That is 1. So when both of them are recessive and here both of the traits are dominant, and in these two, one is dominant, another is recessive. Similarly, one is dominant, another is recessive. Well, so this is the ratio 9:3:3:1 that we are supposed to get. Now the question is asking the calculated percentage probability of dwarf plants with red flowers. So the dwarf plant and red flowers, that means this particular type of phenotype is asked. So it will be 3 out of 16. So the probability will be 3 out of 9 plus 3 plus 3 plus 1, that is the total. So it will be 3 out of 16. If we calculate, it will be 0.1875. Now here it is asked the percentage probability, and the question is asked in percent. So therefore we have to and also two decimal places. So we have to calculate it to percentage, and for that we will multiply with 100. So it will give us 18.75%. So that will be our answer. So the answer is 18.75, rounded off to two decimal places. Okay, so that is the answer for this particular question. And for more information on the Mendel's genetics, the Mendel's monohybrid and dihybrid cross, you can follow this NPTEL course. The lecture is Mendelian and non-Mendelian genetics. There are 3 parts, part 1, 2, and 3. So that's it. Thank you.